Okay, let's get started. It's 101. Hi, everyone. Welcome. Thank you for being here today. Today is the third day of our 2024 National Postdoc Appreciation Week. And like every year, we always host a virtual career panel. This year, we're proud to have our following panelists, Dr. Paula, Dr. Cardetti, Dr. Gour and Dr. Shrivastava. Thank you so much for taking the time out from your extremely busy schedules to come and talk to our postdocs about your individual and unique journeys. And also, we hope that our postdocs get inspired and get more knowledge about the different kind of career paths that they can take. Uh, whether being basic scientists in academia or in industry, or follow Dr. Pala and Dr. Cardetti and then do a non-typical traditional scientific role. So I'm going to hand over the controls to Dr. Pala. And just for our audience, we'll have each panelist will be getting 10 minutes, and then the last 10 minutes will be there for question and answers. When you want to ask a question, please feel free to either type it into the chat room or just raise your hand, and then we'll be monitoring that, and then we'll call your name out to ask the questions. So, Dr. Pala, can you please share, give your kind introduction?
Second. Can you guys hear me okay? And can you guys see my screen? Okay. Is it in the presentation mode? Okay, perfect. So, hi everyone. I'm Dr. Zarna Pala, and I'm the Assistant Director of the Biological Sciences Graduate Program at the University of Maryland, College Park. And I'm here to share a little bit about my career journey and also share some career tips and advices as to how I made this transition to this position after being a bench scientist for over 15 years. So let's dive into it. Okay. So I started my journey back in India. I'm from a small town called Jamnagar, Gujarat, located on the west coast of India. And I did my schooling from there. I moved to pursue my bachelor's in technology with a major in biotechnology to Navi Mumbai from Dr. D.Y. Patil University. And then I went on to continue my master's in technology with biotechnology as my major from the same university. And I was the gold medalist of the batch. Following that, I went to pursue my PhD in molecular parasitology from BITS Pilani campus. And I was awarded two prestigious fellowships to pursue my PhD, one being the DST Inspire, the second one being the CSIR Senior Research Fellowship. And after I finished my PhD, I moved to the United States to pursue my postdoc research at the NIH. And I also came to the NIH with my own fellowship called the Malaria Research Postdoctoral Fellowship. And I was here on a J1 visa. And after spending 5 fruitful years at the NIH, I transitioned to my current role. And I would like to highlight throughout my journey that I was interested in science and in research from like my school days. And I decided that I want to pursue research. And so throughout my undergrad and my master's, I was actively involved in research, and my field of research was plant biotechnology then. I slowly transitioned to molecular biology during my PhD. And then, again, for my postdoc, I was again still in the field of malaria overall, but then I moved to vector biology. And so I tried to keep my a common theme throughout my research career, which was malaria. Even if I was working in plant biotechnology, I was looking for novel anti-malarial compounds. during my PhD, I was looking for, say, for example, novel drug targets for the treatment of malaria. And in my postdoc, I found out a novel transmission blocking vaccine candidate for the treatment of malaria. So the overall theme was same, but I kept changing, the skills and the research skills. And the other thing which I want to highlight is that throughout this time, throughout my PhD and my postdoc, I was actively involved in a lot of outside the lab activities, which are organizing conferences, organizing workshops, organizing seminar series, inviting speakers, etc., etc. And so I think throughout my PhD and postdoc, that is almost a span of like 11 years, I was also getting and developing my transferable skills, which, sort of helped me to get to this current role. So a little bit about the career decision process in general. People often ask me, you were a scientist, a bench scientist for over 15 years. How did you decide to move to a completely non-bench path? And are you happy with this non-bench path? And my answer is yes, I'm very happy. And I don't regret even for a bit that I'm not on the bench anymore. But I'll go through the career decision process. So I think the key thing is to analyze your skills, your interests, and your values. And your ideal career will sort of fall in this small, junction where that would be your ideal career, which would give you happiness and, the job satisfaction at the end of the day. And I've had I've added like a few QR codes on top sides of all of these skills, interests, and values. And these QR codes will direct you to links where you can actually take these tests. So you can take skill assessment, values assessment, and interest assessment and see what your skills are and what your values and interests are. So this will sort of like give you a broader picture of, and sort of direct you in that path of what you are good at. And then you sort of move to focusing on all these and developing transferable skills. And so usually when I ask postdocs what your approach is when they get to the job market, they would say like, oh, I'm just looking for a job. I'm applying, I'm sending my resumes to 100 different companies. And then I'm like, that's not the way you do it. So you have to start planning for it and make a conscious effort and strategically plan your next move. So you don't have to take the top-down approach, but you have to take the bottom-up approach. And you have to start from skill assessment. And then you go to the exploration of what your SVI results are, who are the people who are doing this sort of job, where are they located, whether they're in industry, academia, government, etc., and how did they get to this job. And this you will get to know by doing informational interviews with different people. So do informational interviews. I would say I did more than 15 informational interviews before, deciding that I want to get into this role. And then you start focusing. So once you've decided that, okay, you want to get, into this role, then you start developing the skills and developing your resume for that sort of position. And then finally, say in your third year and fourth year, you start applying for jobs, networking, interviews, etc. And for all the international people here, immigration is something which you need to think from day one and not in your fourth year. So, prepare your profile from day one, start preparing and start working on it. And I've given a timeline on the right side as well for like when to start this along your postdoc journey. And more sort of like discussing what my current role entails is that I am a PhD trained scientist. And on a day-to-day basis, I do science administration. I run a program which is over 300 people. on a daily basis. I do data analysis, management of the alumni database, tracking the progress of of all our PhD students. I'm also actively involved in like outreach and recruitment and, advertising our program, going to conferences. Um, Again, actively involved in admissions. We organize two big recruitment events every year, so I'm a part of that. And the most favorite part of my job is professional development and career advising and mentoring. So I hold and I conduct workshops for our graduate students to help them, move to their next step. And I also do one-to-one career advising for all our students and help them navigate through the difficult journey of, PhD and beyond. And to all of you who are here in this meeting today, if you are a postdoc, you can get into any of these and beyond this, like careers. It is not just the traditional academia industry. There are things beyond that. You have options in science administration, education, writing, science policy, IPR, patenting, law, business, consulting, and these are only to name a few of them. And all of these exist in all the three major sectors of academia, industry, and government. So, think beyond the bench. There are countless options. And yeah, take charge of your career and professional development today. Do not wait for it. It's never too late to start. And I have a small QR code on the top that is the link to my LinkedIn profile. Please feel free to connect with me. And yeah, if you want to do an informational interview, feel free to reach out. Thank you so much.
Great. Thank you so much, Dr. Paula. And again, we'll open up the questions later on towards the end. I just want everyone on the Zoom to be aware that the session is getting recorded and it will be available online. And then also, the LinkedIn profiles are already out there on the panel. But yes, please feel free to, if anyone has a QR code, please feel free to scan that and get in touch with the panelist. So I'm now going to hand it over to our next panelist, Dr. Cardetti. Dr. Cardetti, if you would like to share your screen. Thank you so much.
You can see the right screen, correct? Okay, perfect. Hello, everyone. I'm Caitlyn Cardetti, technically doctor, but I very rarely use that title. So I'm here to talk about my main role as an editor for BMC Biology and BMC Immunology. But I also serve as a part-time faculty member at a community college. So if you have any questions about teaching on the side as well, I'm happy to answer those. So I recently left my postdoc at Thomas Jefferson earlier this year to start this position at Biomedical Central, where I serve as an editor for two journals, BMC Biology, which is a very broad biology journal. We cover any topic in biology, and then BMC Immunology, which is obviously a lot more focused. So as for my path, how I got there, let's back all the way back up to undergrad. In undergrad, I was very interested in biology. I majored in biology. I also majored in psychology. While I was there, I worked in a hospital as an ICU technician, so I got a lot of clinical experience. And I did a lot of outreach, and I really realized that I love sharing my joy for biology. So I initially thought back in undergrad, I wanted to go to grad school to primarily teach at the college level. I truly enjoy being part of academia and part of the college life. So then I graduated from my bachelor's degree, and I went off to do research at Columbia University, where I was a research assistant for a couple years before I went to grad school That research was more clinical focused than bench focused. So when I initially started grad school, I started as a master's student because PhD programs were unsure about taking me in without having a lot of bench experience. And I obviously had a very heavy clinical background. So I started as a master's student at Stony Brook University. They were happy with my year there as a student that I didn't actually finish my master's degree. They allowed me to transfer into the PhD program, where I then spent an additional 5 years under my advisor, Dr. Daniel Bogenhagen, working on mitochondrial gene expression. While I was in grad school, I focused on building a lot of other skills beyond just my research, since that wasn't my main end goal. I was granted the opportunity to partake in the Alan Alda Center for Communicating Science, which is part of the Stony Brook University campus. They taught me a lot of different skills, both formally through coursework, but also they had a lot of opportunities just to participate in various outreach and communicating to the layperson in general. I also, while I was at grad school, was encouraged and supported to pursue my teaching goals. So Stony Brook University has the IRACTA program, which is a special fellowship for postdocs, where the training grant covers their stipend so that they can have teaching opportunities, and then your PI won't be upset at you because he's not paying for you anyway. So while I was still a grad student, not a postdoc, they allowed me to participate in various programs with them, including this scientist teaching science course, which was a nice pedagogy course that also, if you think about it, teaching is really focused on communicating. Also, while I was there, I did various outreach programs with high school students. I was allowed to teach various lectures while I was there on a variety of topics, and I co-instructed a science policy course. Then fast forward to my postdoc. I was only at my postdoc for about 1.5 years. I initially was still thinking my end goal was to primarily teach. While at my postdoc, I concurrently had got my part-time STEM faculty job at Bucks County Community College. So I was given a whole course to teach myself. I was teaching Biology 101 to non-majors. I was responsible for the course design, the assessments, the lectures, the labs, the entire course. And it was a great experience. As you can see, I'm still there and I'm very happy with it, but it also taught me that I do not want to teach 4 courses every semester for eternity, which is what I initially started grad school for. So I realized I wanted to be able to keep my teaching role as a very part-time gig. It allows me to have the energy to show up for my students and teach in the manner that I want. But it then made me have to reevaluate what I wanted my primary career to be. And in some ways that was easy. I had already ruled out that I didn't want to do research from long-term from the start. It was always a means to an end. So I ruled out an industry bench position. And I started to think about what my values and my priorities are, like Dr. Paula really stressed in hers. I actually worked with a career coach for a little bit, and we had a lot of back and forth discussions about what was important to me, what was not. One piece of advice that really stood out to me was she asked me to focus on my day-to-day and make a list of tasks that I really enjoyed doing and tasks that I really did not like doing. So no surprise, I did not like picking up the pipette, but things I did really like was reading. I love reading about biology, not just my niche interests of mitochondrial biology, but the field at large. I liked advising and helping the high school students, the undergrads, the grad students in the lab think about methodology and the best way to set up their experiments. But from a very hypothetical standpoint, I really did not want to touch that pipette. And then we kind of realized a great fit for that would be an editor role, because that allows me to read a lot of science and to think about methodology and really be part of the quality control process, but not ever touch a pipette. So that leads me into my current role. So giving you a little bit more background on what I do on a day-to-day, I have a stack of manuscripts I read through every day. I am not editing them. A lot of people think I'm editing them for grammatical errors or this or that. I'm actually more of like the quality control, the gatekeeper. I'm assessing immediately if I want to send that manuscript out for review or if I want to desk reject it. So to determine if I want to send it out to review, I make sure it fits the aims and scope of my journal. I make sure that it fits the right impact novelty level for my journal. So BMC Biology is a selective journal, so we are looking for a certain level of impact, whereas BMC Immunology is non-selective. So as long as the science is done properly, I will consider it and send it out to review. So if I move it on to review, I'm the one responsible for determining the right expertise to handle it. I either ask one of my external academic editorial board members who are in the area to handle the manuscript for me, or I myself will handle it. And then I pick out reviewers. I assess their feedback. I decide if I want to have the authors revise their manuscript. If the feedback is, it might help me decide that the manuscript is not the right fit for us, and so forth. I also work very collaboratively with a lot of teams at BMC. So BMC is part of Springer Nature at large. And I work with a research integrity group. I work with a productions team. I work with a whole slew of editorial board members that are external to the company. There's a lot of interaction with other people. And then I also do a lot of journal development. So I reach out to people for commissioning. So most reviews you see in journals are not just submitted reviews, they're invited reviews. So I determine who I want to invite, what topics I want to be inviting reviews to be about. I go to conferences so I can stay up on the latest in the field, and I interact with a lot of people. So if you're interested in being an editor, I also am happy to do a more private editorial, informational interview. But a few things that you need to be good at is you need to be very self-disciplined and able to manage your time because you are mostly working independent. So I have an office, but I also am often working at home. I'm setting my own I work 9 to 5, but I'm setting my own list of tasks I have to get to every day, my own list of priorities. And in some ways, it's a lot like research. Your work is literally never ending. But unlike in the research field, at 5 p.m., I close my laptop and I let whatever is there wait for me till tomorrow. But it is somewhat of a customer service-based position. You don't want to make authors waiting too long. They will send you angry emails. So you have to be very good at communicating with people and making sure you're timely for them. And networking is important. So if you are not a social person, I don't know that I would 100% encourage it. Maybe then you might want some of the more back-end editor roles, like actually copy editing. If you are not good at your own time management, I don't know that I would recommend this position either. And if you don't love reading and reading a lot and a lot more, I also don't know that I would recommend it. But if you like all of those, it's definitely a very fun position in my opinion, and I'm very happy here. So I guess I'll stop there.
Great. Thank you so much, Dr. Cardetti. So I'm actually going to hand it over to Dr. Gour, but I will be sharing my screen for his. There you go. Sorry, give me 1 second. Don't seem to.
Just meantime, I just want to confirm. Can you hear, all of you hear me?
Yes.
Okay, thank you.
Sorry about that.
Thank
It's
you,
not
Ilham.
a proper Zoom call till you don't have some technical error.
Yeah, thank you so much.
No worries.
I really appreciate the Jefferson Postdoctoral Association to inviting me for the one of the panelists. And as I am working in Mount Sinai as a recently promoted as a faculty, I did my master's and PhD and postdoc in India. Can you change the slide, please? Yeah. So I have done my master's, my PhD and postdoc in India within prestigious institutes and with a lot of different kind of fellowships and scholarships. And my master's I did in Central Drug Research Institute, CDRI, Lucknow in India. And my topic was purification and characterization of DNA topoisomerase 2 from filarial parasites, its effect of inhibitors and antifilarials. I purified and characterized DNA topoisomerase 2 from different kind of filarial parasites. And I did the first line screening of chemotherapeutic agents to develop drug against microfilarial disease. I also constructed genomic and cDNA libraries of the microfilaria. And then I did then I moved to my PhD, that is actually in animal biotechnology. And PhD, I got 2 scholarships. My first graduate scholarship for first 3 years from the Indian Council of Agriculture Research, Ministry of Agriculture, Government of India. And then my senior research fellowship from the Council of Scientific and Industrial Research, CSIR, SRF, that is from the DST, Department of Science and Technology, Government of India. In my PhD, I identified and screened some novel gene variants in terms of SNPs and indels to develop an SNP panel, so it's called SNPs panel, to determine synonymous or non-synonymous mutations, to utilize a technique with molecular cloning, DNA sequencing, high resolution melt curve analysis, and Taqman assay through the qPCR. I generated baseline data for the association studies to establish a breeding program based on the marker-assisted selections. And we developed some SNP panels to identify in the beginning the milk and the meat production genes and identify the animals in the beginning before to grow. And then I moved to my doctorate, my postdoc fellowship, and I worked in postdoctoral fellowship in Center for Cellular and Molecular Biology in India. This is a little bit different from my field. Definitely, I did the work in the genetics, but this the most of the work I did in my postdoc that was on tiger conservation genetics. So that I worked in and my work was focused on optimizing and non-invasive genetics tools. And for the first time, I evaluated genetic diversity, population estimation, relatedness, individual movement, demographic parameter, habitat connectivity, assessment of metric system, and behavioral ecology of tiger. Definitely, I used microsatellite markers, mitochondrial sequencing, and qPCR. And I also did successfully lead several wildlife field expeditions, including the biodiversity hotspots across Indian subcontinent, including India, Nepal, and Bhutan. And in my postdoctoral research, I have published several research articles in there as a leading author. And and my impactful work on the tiger conservation has attracted numerous attention by the media, social workers, as well as the government. And that helps me a lot in establishing me as one of the renowned tiger conservation genetist and the youngest tiger conservation genetist. And later on, my research journey moved to what's called in the USA, and I was in the University of Idaho. I was working in University of Idaho as a research scientist, and I was working in two different departments. One is Genomics and Bioinformatics Core, and one is Department of Fish and Wildlife. And I optimized and developed assays for it's called next generation sequencing, especially Illumina and PAGBio and molecular diagnostics. I was the part of the prestigious SARS-CoV and COVID-19 research team in the time of pandemic, and I used my NGS expertise to detect different strains and mutations of the COVID-19 to screen the contiguous variations of the COVID-19. I also utilized my expertise in amplicon sequencing and red sequencing to understand how environmental variations influence biological diversity over space and time. And there's again one great thing happened while as I was working and as a research scientist in the University of Idaho Genomics Core and other departments. And the lab I was in charge and achieved the University of Idaho the top spots called ranked #1 lab in the campus wide. And because the University of Idaho decided to conduct an unannounced lab survey inspections called in around more than 250 laboratories to determine the current lab safety compliances. And the lab I was in charge became lab #1, and that gave me lots of the recognition. And I worked in COVID-19 and as a research scientist in the genomics core, and that helps me a lot. And I have been honored with the University of Idaho's highest award. It's called University President Medal for my exceptional contribution to my pandemic response. Then I moved to Icahn School of Medicine as a senior scientist in Mount Sinai, New York. And I was working since here, and I am really thrilled to be part of the phenomenal team that's revolutionizing the patient care and expanding its reach to the pharmaceutical industry. Here we focus on using our omics expertise as one of the tools to develop cutting-edge drug combat inflammatory skin diseases such as atopic dermatitis, alopecia, vitiligo, etc., etc. And I recently joined Icahn School of Medicine as a faculty as well. And other than my research journey, I was faculty in different countries. I was assistant professor of biotechnology in Jaipur National University, Jaipur. I was assistant professor in Tecnologio de Monterrey and Mexico. This is one of the largest university of Latin America with 100,000 students and 40 campuses around South and Central America. I recently also joined as a faculty in the Icahn School of Medicine. So this is my research journey from my master's, my PhD, and my postdocs to the research scientists and the faculty. And I just want to suggest there are only two things that you have to decide before you want to plan anything after your postdoc. First, you need to decide what's your future plan. Do you want to go to academia or do you want to go to the industry? Because there are two important things that you need to know before you plan in any of the field. That is, first is job satisfaction, second is the job security. So job security is most important, and definitely job satisfaction equally important. So if you are immigrant, you need to know where you get the job security, and later on you also plan where you get the job satisfactions. So if you are planning to continue as a scientist, as a faculty, you have to have mastery on some of your important subjects or some of the important research areas, and then later on you can decide where you want to go. And another thing that is important, other than the technical mind, innovations, knowledge of the field, you have to choose some important ideas and some important field that you can get the job pretty easily in the academia as well as into the industry. So if you have some core competency in the microscopy, genomics, transcriptomics, bioinformatics, flow cytometry, pathology, stem cell research, immunology research, and other kind of aspects, definitely it is a great opportunity for you to work on those kind of fields, because you can get industry jobs easily in those fields, as well as you can get very job security kind of jobs in the different research and course facilities for course facilities in the academia, like industries and medical college and other hospitals. Thank you so much. I have done my part.
Great. Thank you so much, Dr. Gour. I'm going to hand it over to our last panelist, Dr. Shrivastava.
Can you see my screen?
Yes.
Yes, sir.
Hello everyone, my name is Gaurav Shrivastava and I am a research scientist as a research fellow in National Institutes of Allergy and Infectious Diseases at NIH in Maryland. So I start my professional journey from master's due to time crunch. So I did my MTech biotechnology from Vellore Institute of Technology from India, and my master project I did from CCMB Hyderabad, India, and where my project was molecular characterization of NEF gene of HIV-1 from clade B and clade C, where I learned the basic molecular biology and virology. And after my master's, I did my PhD in the Department of Molecular Medicine at Shrivastava, Mexico City, Mexico, where I work in two different projects surrounding dengue virus, where my work is to identify the dengue virus genome, where is some of the protein work as a viroporin, and if there are viroporin, what are the characters and what they have role during the infections. And other aspect, I work in the innate immune response of the dengue virus, where I work to understand how the inflammasome is activated during the dengue virus infection and what is the molecular mechanism behind it. After finishing my PhD, I moved the first postdoc at University of Alberta, Canada, where my project was surrounding on brain tumor glioblastoma multiforme, because in the brain tumor, generation of glioblastoma is also caused by one of the inflammasome gene, as well as the HIV infection in the brain. As we know that during the HIV infection, HIV crossed to the blood-brain barrier, and there also it has been shown that HIV caused the inflammasome. So my work is to understand how the HIV is causing the inflammasome, and due to that, the exons are getting dead. And whatever drug we have till now, they are not crossing the blood-brain barrier. So it's very important to understand how to stop the inflammasome in the brain and stop the damage of neurons. However, after just 6 months of this postdoc, I understand that this lab was not for me, because I feel that it was not very welcoming, and also it was giving me a lot of toll on my mental health. So after 6 months, I understand that okay, I need to move on from this lab. It doesn't matter how good is the project. I was very enjoying the project, but I say, you know what, I cannot see myself more than 5-6 years here. So I started looking for the lab, and then fortunately I landed in the laboratory of malaria and vector research at NIAID NIH, where I continue my work surrounding the inflammasome, where I continue the work with the dengue virus and human host, as well as in the NIH, I work with the triad, where I wanted to understand the role of host as a human, virus as a dengue virus, and the vector as a mosquito. So I just wanted to understand what is the role of mosquito, virus, and human in the activating the inflammasome. As we know that during the mosquito bite, mosquito release the virus in the skin, and it has been shown that there are several, I identified several proteins in the mosquito saliva that actually help the virus to infect, as well as also to mitigate and control the inflammasome and causes the severity of human disease caused by the dengue virus. During all my postdoc work, I experienced both the excitement and the pressure of deep scientific research. The excitement of discovery fueled my passion, but the reality often involved long hours, the pressure to publish, and uncertainty of job instability. It was very rewarding yet a challenging phase where the persistence was a key. And one turning point I can say came during my particular intense period of research. I was coordinating multiple projects. I was mentoring several junior students, as well as participating and handling several postdoc committees. It became very clear to me that while I love the science, I was drawn to the organizational and leadership aspect of running a lab. This realization sparked the idea of exploring career beyond the bench, where I could make a broader impact on scientific progress. Therefore, and then I started looking for a transition. And in these weeks, coming weeks, I'm transitioning permanently to the academia to science administration, where where my transitioning from postdoc to research fellow to the scientific operation manager was driven by a desire to create a broader impact beyond the bench. I realized that I love the research, but I wanted to contribute to science at an organizational level, that helping labs in running smoothly, facilitating the collaborations, as well as the ensuring resources are used effectively. This shift me to focus on operational efficiency and contribute to the largest scientific mission at NIH. I also sought a work-life balance, which is often very hard to find in the very demanding research career. This desire to have a broader impact, as well as balancing my personal and professional life, prompted me to consider to transitioning into scientific operations role. What sparked the change I mentioned, and what is the role of my PI? Fortunately, my PI has a very significant role in my career trajectory. I can say my PI has a true leader who allowed me to pursue my interest and showed the path to me to become successful and support my activities outside the lab. And we had very active and positive communication about our mutual interest, and discuss in detail my path to do my transitional very smoothly and successfully. What are the skills transfer that I got it? So many of the skills that I owned during the PhD and postdoc are smoothly transitioned to the new role. For example, problem solving role that as a PhD and postdoc we use daily in our lab, becoming very essential for me in addressing logistic challenges. And another like project management, and that is a is very natural part of the running experiment, and several coordinating research that help me handle large initiatives involving multiple teams. And perhaps most importantly, the communication skills that whether through scientific writing or showing your research in the conferences, it proved very invaluable skills that I began working with the wide range of stakeholders from researchers to administrators. And these skills laid the foundations for the success in my new career path. What are my contribution? So as a coming from the biological background, I bring a unique setup of skills. I understand the challenges that researchers face, allow me to serve as a perfect bridge between the researchers and the administrative and operational aspect. And it helped me to advocate for the need of the scientist while ensuring operations and aligned with broader organizational goals. What are the challenges I faced during this transition? I can say that one of the hardest part of the transition was stepping away from the bench that I have done from last 16 to 17 years. It was also challenging to adopt a managerial mindset, shifting from focusing on experiment to focusing on people, budget, resources. This learning curve with understanding administrative process and managing diverse team was really a steep. And learning and growth. To overcome all these challenges during the transition, I relied heavily on networking and mentorship. Talking to others who had had a similar transitional helped me understand what to expect and how to prepare. Leadership development program also helped me to adjust to manage the big teams while learning about budgeting and resource management enable me to navigate the administrative aspect of this role. As you can see here, there are several roles that scientific operations manager do in day-to-day life at NIH. But as a brief, scientific operation manager at NIH plays a crucial role in ensuring that labs and research facilities operate smoothly. My responsibility include overseeing daily operations, managing budgets, and ensuring teams have the resources they need to succeed. I'm also involved in the strategic decision making that impact everything from hiring to resource allocation, resource management, safety, biosafety, chemical safety, lab safety, operations management, SOP development, and liaison between departments and central NIH. And I have several, some of the recommendations for the postdoc here, that first of all, I would like to emphasize that please enjoy the path rather than having anxious about getting a transition or getting a different role. First, understand that one of the thing I learned, that the career path in science are not linear. There are many ways to contribute to the scientific community beyond traditional research roles. Being flexible and open to explore new opportunities is key to finding a path that align with your both strength as your passion. And as Digpal as well as Zarna say, and Caitlyn also, to define professional goals. Consider that the postdoc is very temporary positions, and make sure to have a schedule, regular meeting with PI to discuss both research and professional development. And please focus, I would say, please focus to develop your soft translational skills, focus on developing communication, leadership, networking, mentorship skills, as well as regularly communicate science finding through presentation and written reports. Take leadership role from the lab, start from the lab, delegate tasks and mentor less experience your juniors, and also take the leadership role over there. As well as it's very important to seek out multiple mentors from various sources. It can be from LinkedIn, it can be from your university, it can be from Twitter, but start networking as soon as possible. And once you do it, please go ahead with the informational interviews. I have done several informational interviews, and then I got to know that where my niche is, what I can do, where is my strength, and what I need to expect, and what to do and what not to do. And I would say, please connect with the institute training office, your postdoc office, and explore available resources for career development. Always seek advice from your career counselor in your postdoc office, and enhance your training experience. And most important part, during all these things, please take care of yourself. Having enough sleep, engaging in activities outside the lab, as well as please make sure to celebrate the achievement, whether it's small and big. With that, I would like to say thank you so much. And if somebody wants to know more about my job in details, or the job that NIH offers after postdoctoral positions, I am very happy to reach out. Please reach out to me by email, LinkedIn, or Twitter. Thank you so much.
Great, thank you so much, panelists. That was a really helpful information. I'm just going to open up the questions by asking a couple of generic questions. Well, not generic, but so that we have an opinion from all of the panelists, and then we'll move on to the questions that people have been writing in the chat, and then Joyce will take care of that. But one of the main takeaways that I'm getting from all of the panelists, and you guys, the panelists can, feel free to unmute themselves at any time and chime in. Also, I can see your screen, so maybe I'll call you guys out if you want to raise your hand. One of the important takeaways that I got from all of your messages was that networking is important. And then once you figure out what your skills or what your niche is, feel free to go ahead and do a lot of informative interviews, because that is the way that you postdocs or students will kind of figure out what a day-to-day looks like for that particular job that they're trying to apply for. Is that correct? we got a nod, so that's pretty much it.
Definitely. Yeah.
So I think one of my main questions that I think is also like a very common theme is that how soon would you start approaching your PI with the career conversation, and then how would you actually approach? What would be your suggestions to your panelists how to approach your PI, and then start saying like most of you have actually gone for non-traditional bench work, and some of you are still in the bench position. So let's start with that question. Zarna, do you want to start first?
Sure, yeah. So I think I would start with day one. I started with day one when I started as a postdoc fellow in my with my PI. On day one, I told him that I did not want to become a PI, and I want to transition into something which is more translational and non-bench. And so my advice would be to have those candid conversations from day one, but you also have to test the waters. So be very mindful and see what history, says with the particular PI. So if the PI has historically not been very supportive about any transitions or change, then, of course, be careful. But like in my case, my PI was very supportive. He also actually did a mock interview with me when I was applying for this job. So he was very supportive. He looked through my resume, my cover letter, etc. So I would say day one, and not to wait. If you know that your PI is very supportive and you can have that candid conversation, then start from the very beginning so that you can, start building your resume for your dream job, because you will have to do a lot of things outside of the lab if you're looking to transition to, a non-bench position. And so your PI's approval would be like the number one thing you would want. So yeah.
Great. Also, there are some specific questions for each panelist in the chat. I want to be able to answer everyone's question. So I'm actually going to send it, sorry to cut this question a little bit short. Thank you so much for your answers, Dr. Pala. Joyce, do you want to start asking the questions in the chat so that we have all of our, participants get their question across?
Yeah, sure. There is one question from Shushrat. It's what was the most important factor that you would consider for career? How much did lab environment versus the topic of research played in your decision to pursue each of your respective paths?
Does any one of who shall I just call on Dr. Cardetti? Do you want to answer his question?
Sure. Self-awareness, know yourself. As Dr. Paul, like you want to know your values for your career choice. So I think we all think we're pretty self-aware, but we probably could all use work there. So if you need help with reflection, like I said, I found a career coach very helpful because it was something we could go back and forth about, and they can ask you questions that you might not probe into yourself about. But there's also great resources, like there's a book called Pathfinders that's very helpful if you don't want to work with a person, but you got to do the work for self-awareness. Whether research topic or not matters. It depends what you want your end goal is. So for my role, research topic doesn't truly matter, but I would say that when I was being hired, they were appreciative of my background in the mitochondrial field, since that is a hot topic. And they also appreciated my background teaching at a general biology level, since I handle manuscripts in all categories. So I have both a niche focus, but can serve as a generalist.
Great. Dr. Gour, do you have anything to add to that?
Joyce, can you repeat the question? Sorry.
Yeah, I can read that question again. Give me 1 second. What was the most important factor that you would consider for career? How much did lab environment versus the topic of research played in your decision to pursue each of your respective paths?
Okay, so there are two things. One is the lab environment. Another is your own expertise. So it depends actually. So lab environment is supportive. That really helps always. It doesn't matter. It's in your professional life, your personal life, or your any other life. So that is always important if you are working in a very good lab, or working in a toxic lab, or working in any other lab. So that is always important. Doesn't matter. You are going to. It helps you choose your career path, or it will help you in support in your personal life or your any other life. But definitely, the second thing, that is your research topic. That is equally important, because this research topic in the future choose what you are going to do it, because there are very limited scopes in the future when you can get any kind of general job. I mean, the job, it's maybe a little bit related to your past 10 or 15 years of experience. And so definitely most of the postdocs will go to either in academia or go to the industry. And definitely it will help what they have done in the last few years. So the topic is equally important.
Great. Joyce, let's skip to the next question, and then we'll have Dr. Shrivastava kind of give his opinion about that.
Sorry, I was muted. I have one question for Dr. Cardetti. On what basis you decide the impact of paper? In other words, what scientific criteria you apply to rate an author's work as high impact?
Well, I'm sure most of you participate in journal clubs, so you in just being a researcher, you should have a sense of what is groundbreaking in your field versus what is incremental. So when I'm talking about impact, I'm talking about that there. So it's really gonna depend on what journal you're submitting to. Obviously, we often think of Nature and Cell and Science as really high impact journals. That means for you to get published there, you have to have a really novel finding, something that's going to change maybe the field, or something that's just, it's very important. Whereas when you start to look at other journals, they have different levels of selectivity. So for example, BMC Immunology is a non-selective journal. As long as it's good science, I will send it out to review. And what I mean by that is appropriate methodology, appropriate controls. It's ethically done. So if you're doing human studies or animal studies, I care a lot about the ethics. So as long as it ticks all of those boxes, that would be appropriate for publication without it needing to be largely impactful. BMC biology is selective. So for that, I want to make sure the journal, the submission is adding some level of novelty. So to judge that, I can look at the references in the manuscript itself to position myself in the field, because I read manuscripts on mitochondrial biology, but I also deal with all the immunology manuscripts, the microbiome. I read about beetles, I read about pollination. So I have to have a broad sense of a lot, which is a learning curve, but I can see what's currently published in the field. What do we already know about this protein that you are studying? Is what you found kind of like oh, duh, like we already knew it did this in this system, in this system, in this system, and now we're just showing that it also does it in this system. That's not hugely novel or impactful. So it really depends on what journal you're trying to submit to. I would encourage you to look at their current publications to kind of ground yourself in what they're looking for. I don't know if that fully answers it, but it's somewhat subjective. So what helps if you want to get published and make the impact clear is to write a good cover letter and make my job easier by highlighting why it's novel, why it's this, especially if you're submitting to a broader journal. It's a good starting point for me.
Great, thank you so much. Just very quickly as a last question, and since Dr. Shrivastava hasn't gotten a chance to give his opinion, Dr. Shrivastava, we have a question here. Do you have any tips to advocate for yourself about projects, career transitions, or professional needs?
Can you please repeat the question?
Repeat it again, sorry about that. Do you have any tips to advocate for yourself about projects, career transitions, or professional needs? So like, how would you suggest that someone goes around like trying to self-advocate for themselves, saying that, you know, they have a specific skill set that they would like to use that for career transitions, you know, and towards like other professional employment opportunities?
Yeah, so as I mentioned in my talk also, start from your lab. Take the leadership from the lab. Do the mentorship in the lab, and then when you do it, go for the whole department level and show yourself that you have those skills to take care of the whole committee, right? And once you are doing it, people keep seeing you, okay, you have this skills, and then you go for the big communication from the like you guys are doing it, right, as a leadership, as management. And once you are doing it, and then take more experience course, like go for the LinkedIn learning courses for leadership, management, and empathy courses that will really help you to focus yourself, as Zarna also mentioned, to make sure that self advocacy is very, very important to promote yourself in the social media. So once you have those skills and you have shown in the department and in your institute, go ahead and keep applying the jobs.
Great, thank you so much. Unfortunately, we are out of time and we're actually slightly over. So I just want to thank again the panelists for again taking the time out from their schedules and like talking to us and walking us through your amazing scientific journey. Thank you all to the participants for coming in. I hope you guys all got inspired. And like I said, this was the third day of our National Postdoc Appreciation Week. Please look out for the next 2 days. Tomorrow is our Postdoctoral Research Symposium starting at 11 o'clock. It's going to be in the Connolly Auditorium as well as the first floor lobby of Hamilton. And then we'll be ending the week on Friday with the scientific reviewing and editing team. So please look out for those 2 events. Once again, thank you everyone for joining in and have a great day. Take care.
Chedam, for the invitation.
Thank you so much
It was
for
wonderful.
Thank you. Bye.
Thank you guys. Thank you so much. Take care.
Thanks, everyone.